All right. Well, good evening, everyone. I'm glad you could be here. My name is Alexa Shavy, and I am a senior academic advisor in the College of Liberal Arts and Sciences Academic Advising Center at Grand Valley. And I am also part of the pre-professional team. You may have also met my colleague, Danny Lauer, in the back there on your way in. Danny is also an academic advisor in our office and also part of the pre-professional team. So I'd like to, again, just say how excited we are to be hosting you in GVSU's newest building, the Daniel and Pamela DeVos Center for Interprofessional Health. This building opened in May of 2021, and it is the third GVSU building on the Grand Rapids' Medical Mile. It features 17 classrooms, 12 interactive laboratories, which are equipped to create interprofessional team environments and encourage collaboration among health profession students. Tonight's event, the Your Health Lecture Series, is a unique educational partnership between Grand Valley State University, Grand Rapids Community College, and Michigan State University College of Human Medicine. The goal of the series is to feature physicians and researchers who can speak on a wide variety of topics of interest to public audiences. Earlier this evening, tonight's speaker met with pre-med students from GVSU and GRCC. And additionally, we had pre-med students who had the opportunity to talk to current med students at Michigan State College of Human Medicine. So thank you to Dr. Campbell and to the medical students for providing that networking opportunity. I'd like to now introduce you to Mark Breavy, the Director of Community and Government Relations from Michigan State University College of Human Medicine, to provide some additional information and introduce our speaker.
All right. Thank you, Alexis. And I want to thank Grand Valley State University for hosting the event in this great facility. This is our second year that we've had this event in this building, and we are just so grateful for the opportunity. This lecture would not be possible without the co-sponsors of the event, Grand Valley State University, Grand Rapids Community College, and Michigan State University College of Human Medicine. This is actually the 10th year of the Your Health Lecture Series event at Grand Valley State University, and this is the 9th year of this partnership with Grand Rapids Community College. So this year we combined our efforts, just as we did last year, into one event due to the unique partnership we have through the Early Assurance Program agreement between our three schools. GVSU and GRCC students are eligible for early admission to medical school through this program, and GRCC students have pathways to the College of Human Medicine, both through Grand Valley State University and Michigan State University undergrad. Now, without further ado, I'd like to introduce our speaker for this evening. Daniel Campbell is an assistant professor in the Michigan State University College of Human Medicine, Department of Pediatrics and Human Development. He earned his PhD at Penn State and has also worked at the University of Pittsburgh, Vanderbilt University, and the University of Southern California. His laboratory studies the impact of genetic and environmental factors on brain development and autism spectrum disorders, ASD. It is widely believed that gene-environment interactions contribute to ASD risk, but there are few concrete examples of specific gene-environment interactions. Dr. Campbell's group was the first to show a specific gene-environment interaction between the gene MET and air pollution, both of which independently contribute to ASD risk. His most recent work focuses on gene-environment interactions that involve air pollution, and his work shows interactions with both well-established ASD genes and novel genes. With that, we'd like to welcome Dr. Campbell.
All right, thank you, Mark, and thank you to everybody for the invitation to come talk. Just as so, you've got all my information in the intro, and I just want to give kind of a broad overview of genetics and environmental factors and how they contribute to autism, and then I'm going to kind of drill down into what my lab does. How do I go forward? There we go. So our goals for today are to to define what the role that genetics play in autism spectrum disorder, which I'm going to abbreviate as ASD throughout the talk. So don't lose that abbreviation. If you see ASD, I'm talking about autism. Increasing diagnosis rates and why high heritability may not be the only factor that increases ASD risk. And then what are some of the environmental factors that contribute to ASD? So that's what we're hoping to get out of the next 40 minutes. Keep hitting the wrong button. All right. So for those of you who don't know, if you showed up to this talk, you probably do know, but quick introduction to ASD. It's a neurodevelopmental disorder characterized by impairments in social communication and behavioral flexibility. So kids with ASD have problems with making friends, keeping friends, interacting in a conversation, and they also have problems with they like to have the same routine over and over again. And if you deviate from that routine, it upsets them. It's now diagnosed in 1 in 44 children. So 2% of the population, pretty much every other classroom in the United States has at least one kid with an autism spectrum disorder. It's 4 times more common in males than in females, and that is a fact that we simply don't understand yet. It's got to be a big hint of what are the underlying causes, but to my knowledge, nothing has developed to explain why males are more often diagnosed than females. There are both genetic and environmental factors that contribute, and I obviously read this literature a lot. Every genetics paper ends with, we found this really interesting thing, but it doesn't really explain a whole lot. There must be gene environment interactions that help to explain how the genetics contributed. And every environmental paper ends with, here's what we found. It explains a little bit, but not a whole lot. There must be genetic factors that make some people more susceptible. The question is how these two things come together. What are the gene environment interactions are widely believed to contribute to autism, but we don't have evidence for very many specific gene environment interactions. And my lab is charged with trying to change that. So this is just getting at the prevalence of ASD. You can see that at the top in the year 2000, that the prevalence of ASD was 1 in 150 kids. It stayed there for the next essentially 6 years. And in the past 12 years, that rate has nearly tripled. So we're now down to, instead of 1 out of 150 kids, so 1 in, every, 5 classes in an elementary school classroom. It's now 1 out of every 2 classes in an elementary school classroom has at least 1 kid with ASD. I'm going to do this now. I'll probably repeat it. Genetics doesn't move that fast, right? So genetics moves from one generation to the next every 20 years. And genetics explaining ASD, this is counter to that. You cannot explain how genetics entirely would drive the rate from 1 in 150 in 2000 to 1 in 144 in 2018. Genetics just simply doesn't work that fast. So what are some of the possible reasons for this increasing prevalence? One is that, and it's true, the diagnostic definition of ASD has changed and it's broadened a little bit, not 5 times, but a little bit. There's more awareness of ASD and among parents and doctors. There were parent groups of children with ASD who did an amazing job taking it from something that honestly you'd never even heard the word autism probably 30 years ago, to now everybody knows what autism is. It's gone from something that was not very long ago thought of as a brain disorder that was caused by the mom not loving their child well enough, to we now know that it's a biological, there are biological causes for it, and that we can hopefully get around to treating those with biological therapies. There's also more willingness to it, right? So at one time, if your kid was diagnosed with autism, they were sent to an institution and that's where they spent the rest of their days. Now, there are lots of people will tell you that their child has autism right up front. It is in many ways a badge of honor that you are helping this kid who's got additional difficulties that not every kid has. And finally, ASD diagnosis provides access to special services. It is absolutely true that there is diagnostic substitution. A very good example is in 1994 in the state of New Jersey, there were 3,000 kids who were diagnosed with something called specific language impairment. In 1995, the state of New Jersey decided that they were going to start providing services for kids with autism. And they were the first state to do that. And that was great, but they had to cut the budget somewhere. So they cut the budget for giving services to specific language impairment kids. So they were no longer getting speech pathology treatment. In 1996, there were 3,000 more kids who had autism, and there was nobody who had specific language impairment because everybody had gotten their diagnosis changed to get the treatment that they actually needed. In some sense, that's a medical doctor's job, right? Is to get the child the diagnosis that will help them get treatment. And so there's absolutely diagnostic substitution from something that may look a lot like autism, but isn't quite. Give it the diagnosis of autism and let the kid get the treatments that will help them. The speech and language pathology, the occupational therapy, the things that will make their lives better. The increasing prevalence, as I already hinted at, genetics probably doesn't do it. Genetics doesn't move fast enough to get you a threefold increase in the rate of autism in 20 years. Environment can, however, but what is it about the environment that's changing? And we don't have the whole answer to that yet. Part of it is air pollution. As I will show you, part of it is air pollution that is tiny little particles. We have regulated air pollution in this country to get rid of big chunks that make fog, right? What we've made cars do is break down particles into smaller and smaller pieces. Turns out that those smaller and smaller pieces are the pieces that are correlated with having autism. And I'll come to that. All right, so what causes autism? Genetic factors. The heritability of autism is about 80%. That is ridiculously high. It's higher than any cancer, higher than any other psychiatric disorder. In fact, we used to define heritability by taking the rate of monozygotic twins both having a disorder compared to the rate of dizygotic twins. So monozygotic twins come from one egg, right? And dizygotic twins come from two eggs, but born at the same time. And we used to subtract those two numbers and multiply by 2. If you do that for autism, you get a heritability of 126%, and 126% doesn't make any sense. So we had to change the math on how we do heritability. But ASD is the heritability is extremely high. The sibling recurrence rate is 10 to 20%. So remember that in the general population, it's about 2%. If you have a sibling with autism, the rate of the next kid having autism is about 10 to 20. So it's clearly running in families, right? You're 5 to 10 times higher risk of autism if you have a sibling with autism. And finally, autistic traits are more common in family members of individuals with ASD than in the general population. But parents, when you test parents of kids with autism for whether or not they're willing to be socially interactive or whether or not they have specific little repetitive behaviors, they tend to have more of those types of things than the general population. So it's clearly running in families. So we started to apply genetics to autism, and I want to take a brief diversion here to the surprises from the Human Genome Project, right? So Human Genome Project, enormous success. It came in much faster than it was supposed to happen and much under budget. We were supposed to be able to, at that time, beginning in the 90s, it was supposed to take 13 years and $3 billion to get a single human genome. Increase the technology to the point now you can get a human genome for about $1,000 in about 19 hours. So a huge success, right? So we can now sequence everybody. In fact, the state of Michigan is sequencing every newborn, right? So in no way, shape, or form was it any sort of disappointment, but there were surprises. One surprise is that our DNA has far more mutations than we would have expected. It used to be that if you found a mutation in a gene and the person had a phenotype like autism, you would say that that genetic mutation causes autism. What we now know is that every person in this room, every healthy person has about 20 mutations in their genome that we at one point would have said was causal for a disease. The 20 mutations that truncate a protein and make you have not enough of that protein in your cells. You have 20 of them. All of us do. So in that context, it's really hard to find what is the causal mutation that's causing autism, because 19 of the mutations in an individual with autism have nothing to do with their autism. And the 20th one does, but which one's the 20th? We don't know. The other surprise is that the Human Genome Project was started because we were supposed to find all of the protein coding genes that cause autism, cause schizophrenia, cause cancer, cause every type of characteristic you can imagine in human beings. And it was supposed to be that we were going to find 150 to 200,000 protein coding genes. We were going to find genes that encode proteins. The surprise was that not every gene encodes a protein. So the reason why I'm showing this slide, if I can get this up, in red is the amount of DNA that is contributing to protein coding genes across a number of different species. And this is from single-celled organisms like bacteria all the way up to humans are out here. The blue is showing you the amount of DNA that your body makes that is not coding for a protein. It's making a non-coding RNA. And I'm going to define that better later, but it's just a gene that gets made into RNA, but doesn't make a protein. You can see in this red, we knew when the Human Genome Project started, we had already sequenced these shorter genomes. So these are shorter genomes, and you can see that there's a nice line that you would predict to go up to here. And that's why we thought there were going to be 150 to 200,000 protein coding genes. Turns out in humans, there are 20,000 protein coding genes, the same number as in fish and fruit flies and giraffes and any other animal you want to name. It just levels off out here. So this gets straight. The number of protein coding genes doesn't change. There is no such thing as a protein that is specific to human beings. Nothing. What is specific to human beings are these things called non-coding RNAs. And here's Homo sapiens. Humans have the second highest number of non-coding RNAs. Anybody want to take a guess of what is higher? No class participation. Yeah. What is so I said that humans have the most non-coding RNAs. They have the most DNA that's dedicated to not making a protein, but making other RNA transcripts. And I'm asking, what is number one on that list if humans are number two? Number one on the list is cows. Cows are incredibly complicated. They've got 3 consecutive stomachs. Takes a lot of regulation to do that. Humans are second. Our brains are almost as complicated as the cow's stomach. All right. So surprises from the Human Genome Project impact we're going to talk about in autism genetics, right? One is that there is no such thing as a protein coding gene that causes all autism. And secondly, we're going to talk that every person is walking around with way more mutations than we expected. And so defining what are the genes that are responsible for autism or any other sort of phenotype, any other characteristic is more difficult. All right. So we did it anyway. We sequenced, I think this is 35,000 people, 11,000 of them with autism spectrum disorder, and found that there are a handful of genes that stand out as having more mutations than others. So on the right here, it shows you that there's 102 genes that are statistically associated with autism. But you can see on the top, there's 3 or 4 of them that really stand out. And we're going to spend most of our time talking about this gene called CHD8. We'll get back to that later. But this is all based on sequencing all of these tens of thousands of people. We're looking for mutations that stop the protein from being made. So they're loss of function mutations. And for these genes, those things happen more often in people with autism than they do in the general population. All right. The other way of looking for genetic variants is what's called genome-wide association study, or GWAS. And these look at there are millions of common genetic variants. So we've already talked about how human genetics is very commonly has variants throughout the chromosomes. Here on the bottom is each of the chromosomes mapped out. As mapping up, we're looking at the higher significance of association with autism. And I will tell you that there are 7 peaks that reach above this red line, which is a designated level of significance that we accept. And there are 7 different regions of the genome that are associated with autism, just based on common genetic variants. So there's the 2 ways we look for things that might contribute to autism risk. But this is kind of a global contribution of those types of genetic risk. And I just want to point out that the mutations that you hear about account for about 10% of the risk for autism. Common genetic variants account for about 40% of the risk for autism. And then there's this big gray slice of the pie that we have no idea what's causing those individuals to have their autism, right? So rare variants explain a very small amount of autism cases. Common variants explain more. There's a big chunk of that pie that we don't understand yet, and we're still working on. All right, so that's what I'm going to talk about for genetics for now. I want to switch to environmental factors. Number 1 environmental factor is parental age. Especially older dads have a much higher risk of having a child with autism. Older moms to some degree, but it's mostly the dads driving it. That could be either genetic or environmental. It could be that older dad has a genetic variant that caused him to reproduce later. It could be that dad's sperm is exposed to more environmental toxicants as he goes along, and that's how they get mutated. The prenatal exposure to one is air pollution. Air pollution, as I will show you data, absolutely no doubt that air pollution contributes to autism. The level of evidence that we have that air pollution contributes to autism is identical to the level of evidence that we have that obesity contributes to type 2 diabetes. There's just no doubt at this point. So air pollution is number 1 on my list. Maternal antibodies to fetal brain proteins. I'm happy to talk to anybody afterwards, but there's really good evidence that moms who have specific antibodies to brain proteins have a higher risk of having kids with autism. And a group at University of California, Davis, led by Judy Vandewater, does all of that work. Prenatal exposures could be that drugs used by mom during pregnancy to treat seizures. Valproic acid is still holding up. Anxiety, thalidomide is still holding up. Or depression, SSRIs, it seems that SSRIs are not going to hold up, but valproic acid or thalidomide, luckily there are alternatives that if you know you're pregnant, you can switch to. You can usually switch to other drugs to treat the seizure disorder or the anxiety disorder. And finally, organophosphates. There's a fair amount of literature. It's not entirely convincing that that pesticides that are put onto crops may be contributing to increased risk of autism. All of that evidence is correlational. However, there's a need for cautious interpretation. And I put this up. I had a paper that was published and got some press. And this is in Time magazine. On one side was about my genetic study. On the other side was, does watching TV cause autism? And it was about the Teletubbies. And the actual paper that they were reporting on was interesting in that if there was more rainfall in a county, that county tended to have higher levels of autism by a little bit. But the conclusion that Time magazine made was that if it's raining more, the kids must watch more TV. Not necessarily true. It could be that there's mold in the environment. It could be 1,000 other things. But all of this is correlational, and you always have to be careful of how you interpret environmental studies. All right, so gene-environment interactions, as I said, everybody thinks that gene-environment interactions are the true cause of ASD, but there's very little evidence to support that. And the challenge is that we've already talked about, it's hard to find what the genes are. You can also think about, it's hard to find what the environmental factors are. There's at least 80,000 chemicals in our environment that we know of. and most of them aren't even measured, let alone try to predict whether or not they contribute to disease. Some examples of gene environment interactions that we know of, this is not autism specific, but things like phenylketonuria, where a kid has a mutation in a specific gene. If that kid eats a diet that involves phenylalanine, they're going to get a brain swelling and very sick and die. If they eat a diet that does not include phenylalanine, they will live a long, happy, productive 70-year life. Other examples are things like response to poison ivy and bee stings. Some people have a very strong reaction to poison ivy, and that is entirely genetically determined. But if you live in a place that doesn't have any poison ivy, that genetic susceptibility will never be expressed, right? So you can control your environment if you know what your genetics are, is kind of the point. All right, so this is where I get into talking about specifically my work. And so I'm going to talk about air pollution. I've already kind of hit the punchline here, but air pollution contribution to ASD risk was first shown in 2013 by Heather Volk to be a environmental factor that doubles the risk of autism. So here, when you see an odds ratio, the odds of being the same as 1, if it's an odds ratio of 2, that means it's double the risk of autism. And in this case, what they were talking about is living within a half a mile of a large interstate. So if you live within a half mile of a large interstate, you are twice as likely if mom lives near interstate during pregnancy, the offspring is twice as likely to get an autism diagnosis. A systematic review of those types of studies showed that it's a little bit more than double. So this is just looking at 3 different studies. And the only point you need to look at is down here, where they're looking at it's about 2.3-fold increase in the risk of autism if you're exposed to high levels of air pollution. And one of the more recent studies is, again, just trying to summarize all of the data. And I told you that it's the little tiny particles that we're trying to regulate, and they're also the ones that are driving the autism risk. This is breaking down by PM 2.5 means particulate matter 2.5. So it's particulate matter that's less than 2 microns. 2 microns are tiny little pieces. This is particulate matter that's very small. And that increases the risk for autism about 2-fold. Particulate matter that is less than 10 microns, so bigger, does not have any effect whatsoever. So it's the very, very tiny particulate matter that's important. And this is a paper that came out very recently that I just want to show. Let's see, just this top row, basically. This is your increased risk for autism. Well, first of all, across the bottom is gestation, right? So the weeks of gestation from 0 to 40. And when you are exposed to the air pollution. And on the top is this PM 2.5, little tiny pieces of particulate matter that's less than 2 microns. It's what we've made car engines make, right? So we make car engines make less air pollution because it breaks down their garbage into smaller and smaller pieces. And if kids or if moms are exposed to high levels of this small particulate matter, especially early in pregnancy, there is a higher risk for autism. And that's true for both sexes. It's especially true for boys. This is one of the best hints we have about the difference between boys and girls. It's not true for girls. So girls do not have an increased risk whenever mom's exposed to this particulate matter. I'm just showing the second line because this is nitrous oxide, which is another part of your exhaust from your car. And there's no real difference in exposure to nitrous oxide and risk for autism. The risk is only with this small little pieces of particulate matter, and especially in boys. All right, so we've covered air pollution and its strong epidemiologic evidence. That drove my lab to look at mouse models. We just exposed mice when they were pregnant to these tiny little particles. We call them nanoparticles, and look for what happens in the offspring of those moms. And the first paper we published was adult mouse hippocampal transcriptome changes with long-term behavioral and metabolic effects. And I'm going to show you this really confusing multi-panel thing next. Don't want you to freak out. We're going to spend 10 seconds on it. This is from that paper, what we did, and we found some interesting stuff about how the hippocampus, a part of the brain, develops, about how the air pollution exposure changes weight and fat, and how the offspring mice regulate glucose. Don't worry about any of those details. But there are changes, and whenever mom is exposed to air pollution, the offspring have differences. The second paper that we published, when exposing moms to air pollution and then looking at the offspring, we were looking for changes in gene expression in the brain that we could correlate to blood expression, so that we could potentially look for biomarkers of air pollution exposure in the blood in humans, since we can't take out the brains in humans, right? So we found some of that. This is just looking at changes in gene expression. The blue are genes that are less highly expressed after the air pollution exposure. Red are genes that are more highly expressed after the exposure to air pollution. And you can see that there are much higher changes in the brain than in the blood, but there are some genes that in both brain and blood go in the same direction. So we do have some hope that we can take blood samples and find out if an offspring has been exposed to higher levels of air pollution. All right, but doing these mouse experiments told us some interesting facts, but they did not change the behavior that you would expect to generally be consistent with autism. So there are some behavioral studies that you can do in mice that approximate what you would see in autism. You can see whether or not mice want to socialize with each other. You can see whether or not mice want to vocalize. Our mice didn't change after exposure to air pollution. We also didn't see changes in gene expression in those genes that we know are involved in autism from our human genetic studies in any of the regions or the blood that we studied. So there are differences between mouse and human brain development that we think make the mouse not terribly useful for doing these types of experiments. A lot of gobbledygook words up there. I'm going to kind of skip over this. I'm happy to talk to anybody in detail. But one is that the cells that make up your brain, the progenitor cells, are different in mice and humans. The migration of some of the neurons in your brain are very different in mice and humans. The gene networks that are activated by activity in the brain are different. They're the things called long non-coding RNAs, which we're going to talk about, and enhancer elements, all very different between mice and humans. And we think that the activation by air pollution that we saw in the mice is just substantially different than what you would see in a human brain. And we're going to get to how do we study the human brain at the end of this. All right, so I told you about MET and how that was a gene that is involved in a gene environment interaction. This is a paper that I published, oh man, a long time ago now, 15 years ago, where we showed that MET is associated with autism. And the only thing you need to get out of this is that MET is associated with autism. There's this marker number 3, which is a genetic marker that shows that there's a p-value of 10 to the minus 6, hugely significant association with autism. We then had a sample of people where we knew their genetics. We had blood samples from them, and we knew their genetics. And we also were able to find out what their air pollution exposure had been, because the Environmental Protection Agency, the EPA, puts up monitors. And so you can figure out, whenever mom was pregnant with this child, what did the EPA monitor say was the air pollution levels? And whenever we did that, we found that the kids who had the genetic variant that's associated with autism and was also exposed to high levels of air pollution were about 3 times as likely to develop autism as kids who were not exposed to the air pollution or didn't have the same genetics. Similarly, for nitrogen dioxide, about 3.6-fold. If you had the genetic variant associated with autism and you were exposed to high levels of nitrogen dioxide, you were 3.6-fold more likely to have autism than neither one of those things being true. So that was our first gene environment interaction that we know about. Again, the air pollution risk is about twofold. Having this MET genetic variant is about twofold, but if you have both that genetic variant and the air pollution risk, it's about threefold. And we now know that if we expose human neurons, so we're getting into the human genetic network, and we're going to do it by growing little mini brains, mini human brains in a dish. If you take cells from humans, you can now force them to replicate what looks like a little tiny human brain in a culture dish, and that's what we're doing here. And we grow these little tiny human brains, and then expose them to diesel particulate matter, which is what comes out of a truck exhaust. And what we found is that this exposure to diesel particulate matter drove down the expression of the MET gene to about half of its normal expression pattern. That's exactly what we see whenever the genetic variant in MET that's present in autism happens. So with the genetic variant, we get half the amount of MET protein. With air pollution exposure, you get about half the amount of MET protein. So you're getting exactly the same outcome, whether it's a genetic mutation or the exposure to air pollution. All right. I'm now going to move on to another gene called CHD8. I showed this before, but again, upper right, these are the genes that have been identified as contributing to autism by loss of function mutations. CHD8 is right there at the top. So it was interesting. CHD8 is something that was found to be an autism gene, and we started looking through the literature, and there's absolutely nothing known about this gene. Absolutely nothing was ever written about the function of this gene. And so my group decided to find out, since we're interested in autism and how brain development is contributing to autism. And so what we did was make a construct that would knock down CHD8, put that into human neural progenitor cells, and did something called RNA sequencing, which just measures the amount of gene expression. And I like to introduce this slide as we weren't the only ones who thought of that. There was the Yale group, the Harvard group, and my group all did essentially the same thing at the same time. And we found essentially the same thing too, that the CHD8 gene regulates the expression of other genes that had already been implicated in autism, but also genes that are involved in neuronal development. All right, so the CHD8 gene is, again, it's the most common gene with a loss of function mutation in ASD. And when we knock it down, we get altered expression of genes that were previously implicated in autism and genes that are involved in neuronal proliferation and differentiation. That suggests that CHD8 is a central hub in alterations of neuronal development that contribute to ASD. The trick is that CHD de novo loss of function mutations, even though this is the highest statistical association we have with autism, accounts for way less than 1% of ASD cases. We're up to, I believe it's 24 people who have mutations in this gene out of the 11,000 that we sequenced. Explains a very tiny fraction, but it helps to get us at the biology of what's going on in altered brain development in autism. All right, I'm going to switch gears just a little bit and very quickly to the central dogma of molecular biology. This is something that I learned as a high school senior in the middle of nowhere, Pennsylvania. My daughter learned this in 3rd grade, but the idea is that DNA makes RNA makes protein. Purpose of RNA is to serve as the messenger, right, between DNA, which is stable in your cells, and protein, which actually does something. It's totally false. RNA does most of the things in your cells, including in the human brain. In the human brain, RNA that doesn't make a protein makes up about 62% of the RNA that's in your brain. This is something we've known for a while. It's specific to humans. There's an increase in non-coding RNAs as you get an increase in complexity. Human brain hits the top. For comparison, in fruit flies, that number is about 20%. So 62% in human brain, 20% in fruit flies. non-coding RNAs drive complexity. In the cells that are going to make human neurons, there's about 45% of all the genes that are expressed are these non-coding RNAs. So things that don't make protein, they're totally involved in regulating how the cell is going to develop. And so I give that to you as a little background because the first study to look for genetic variants that were involved in autism found that there was a common genetic variant on chromosome 5p14, that's associated with autism. It is associated with autism with a p-value of 10 to the minus 10, so ridiculously highly significant to autism. It's between these two genes that are called CDH, don't get this confused with CHD, CDH10 and CDH9. Both of those encode genes that we know are involved that are involved in how neurons interact with each other. So this whole paper was about how this genetic variant must explain those two cadherin genes working in ASD. But when I went to my friends who study cancer and studied this types of genetics all the time, they said, no way, it's too far away. There's no way that it's the cadherin genes. You should look for an RNA that's expressed directly under that peak. And it turns out that there is an RNA expressed directly under that peak. And it's called mucin pseudogene 1. It's an RNA that does not make a protein. So what we now know is that this RNA that does not make a protein is increased in the postmortem brains of individuals with autism by about 12-fold. The cadherin 9 and cadherin 10 genes are not changed at all in postmortem brains of individuals with autism. And similarly, the genetic variants that are associated with autism are correlated with changes in expression of this non-coding RNA MSMP1S, but they're not at all correlated with changes in the expression of either the protein coding genes cadherin 9 or cadherin 10, all of which just tell us that the non-coding RNA is the element that the genetics is trying to point us to, not those protein coding genes nearby. We have absolutely no idea what this gene does. So again, we're gonna make a construct and change its expression in human neural progenitor cells, then look at RNA sequencing. And it turns out that there are thousands of genes that are changed in expression. It's ridiculous how many genes are changed whenever you change this one non-coding RNA. Those are genes that are involved in how DNA packages itself and also involved in how protein gets translated. It's interesting because whenever we looked at the mutations of genes, the rare mutations in genes, those same two biological processes are enriched as well. So it seems to be there's some sort of convergence between those rare mutations and this common genetic variants where we're looking at non-coding RNAs, that it's how protein gets made and how DNA gets packaged are two things that are involved in autism risk. All right, and then I want to look at those genetic things that I just talked about are all influenced by air pollution exposure. And I want to talk a little bit about how we know that. Again, going back to growing our little mini brains in a dish, we now know that if we expose those little mini brains to increasing levels of DPM, diesel particulate matter, again, the same stuff that comes out of a truck exhaust, that we get a decrease in CHD8 expression, which is what you would expect because the mutation in CHD8 would bring down the amount of CHD8 that's expressed. And it also increases the amount of MSMP1S and this other non-coding RNA that I didn't talk about, that are both increased in postmortem brains of individuals with autism. So again, we're getting to the same endpoints of changes in gene expression through either air pollution exposure or through genetic changes. And also exposure of human neurons to increased expression of these non-coding RNAs, these link RNAs, can reduce the expression of CHD8. So this is CHD8 expression on the y-axis in a control. And then whenever we overexpress MSMP1S, we get a decrease in CHD8. So there's this convergent mechanism that's emerging where it all starts with air pollution. Air pollution is at the beginning. Whenever there's increased air pollution exposure, you get an increase in these link RNAs, long non-coding RNAs. either air pollution or an increase in the link RNAs can lead to a decrease in CHD8 expression. And I didn't show you, but it's true that all three of these change what we call neurite complexity. This is how we measure how a neuron is developing whenever we're looking at it at a dish, right? So we can't say that there are different connections in the brain, but we can say that there are different ways that each neuron is sending out its connections to make connections to other neurons whenever we're looking at in this dish. So that's our convergent mechanism of ASD risk. Some future directions that we're looking at. There's now this very cool thing called the comparative toxicogenomics database that was built for cancer. You know, for cancer, we know there's lots of genes and there's lots of environmental factors that contribute to different cancers. But now that we have the list of genes that we know are involved in autism, we can use that database to look at the genes that we know are involved in autism and figure out which environmental factors might contribute there. We're also using something called direct RNA-seq, which will tell us not only what are the RNAs that are expressed and what is changed about them, but also something called post-transcriptional modifications. So if an exposure to air pollution changes a base pair on your RNA so that it's now got an extra methylation or an extra marker on it, we can figure that out by using direct RNA-seq. And the third thing we're doing is transplants of human neurons that we grow in a dish into the mouse cerebral cortex, so that we can keep those human neurons alive for a long time. No, that sounds Frankenstein-y, but don't worry, we're just a few of those cells from humans. We put it into the mouse so that we can keep it along for a long time in an in vivo environment, but then we can go back in and measure what's changed about those neurons after a long time in development. All right, and this is a list of my team. I'm just going to leave that there. The end of my talk. There are people working in my lab now, some lab alumni. Most of the work that I talked about today was done by Stephanie Belinovich and Christy Lewis. Jessica Priestley is actually a medical student who worked in my lab. Mathilde Penta is an undergrad who's now a sophomore who's working in my lab. I have several collaborators at MSU. And several collaborators across the country. My funding comes from the National Institute of Mental Health and the National Institute of Environmental Health Sciences. And one resource that I want to point out is the Autism BrainNet. They're the people who collected postmortem brains from adults with autism. Whenever there was an accident, a car accident, a drowning, a death from an epileptic seizure, they would do the hard work of contacting families and asking if they would donate the postmortem brains, and those are incredibly important to the work that I do and incredibly important to figuring out autism in general. So thank you for your attention, and I think I'm ready to take questions. Yes. Any questions here? Yeah, go ahead. And autism was a correlation relationship. So would you not say that it was causing that? So yeah. So the question is, I said early that the relationship between autism and air pollution was correlation, and she's asking me whether or not I would be willing to call that a cause. Yes and no, right? So I cautioned that everything is correlation, especially in environment. I would caution that everything is correlation in genetics as well. And that we need to do the experiments that I talked about where we're changing exposure to air pollution and through time and through doses of air pollution to see whether or not we get consistent changes. Epidemiology is hard, right? Especially epidemiology where you're looking at environmental factors that are hard to measure, right? Luckily, air pollution, honestly, is one of the environmental factors we look at because there's an infrastructure there. There's an infrastructure made by the Environmental Protection Agency to monitor air pollution throughout the country with I think there's thousands, not tens of thousands, but thousands of monitoring stations. And, you know, for the 80,000 other chemicals that are in our environment, we don't have that as often. To answer your question, I would like to believe that it's causative. I can't necessarily say that. And, you know, to do the experiment is a terrible thing to do. To do the experiment is to expose a mom to air pollution and see if their kid more often comes out with autism. And I think that we're doing that as a natural experiment, but to do it as a controlled experiment is impossible. Yeah. Oh, sorry.
So my question, you mentioned that a lot of the air pollution that is affecting this is those very small particles from traffic.
Mhm.
Is there any, I guess, correlation between the introduction of catalytic converters and this?
Yes, absolutely. Yes, absolutely. Well, so yes, there's a correlation, right? Again, causation, I don't know, but there is clear correlation. We decided that we didn't want to see the horrible smoky fog of things coming out of your tailpipe or out of the smokestacks of coal plants. And we regulated that you must make your exhaust tiny little particles. And literally the regulation is make it smaller and smaller and smaller. It's got to be less than 2 microns. So if you measure how much air pollution of 2 microns is around, it's higher. So there is a correlation there. Yeah.
Do your study include people with a later diagnosis as well, like mainly females who included, did you include as well people who were diagnosed in their late teens and their 20s?
That's a great question. I would have to say we did not go out of our way to do that for sure. So we're just now getting around to what my lab does, looking at these mini brains from different people. So far, we've just used kind of a standard cell line to make the mini brains, but you can get essentially a skin sample from any person and do that. And we're now getting into males versus females, autism versus controls, and yeah, one of the things we're interested in is how early was the autism diagnosis, but we don't have answers yet.
We have a couple that are from the Q&A chat, and they are asking to if you can repeat it. I guess the mic isn't now. So
Okay.
When you talk about the genetic mutations, is this something that would have occurred during pregnancy or during infancy, toddler time frame?
So the question is whether the genetic mutations would happen during infancy or during toddler time frame. The answer is that the mutation would have happened in infancy. One of the tricks that I kind of glossed over is that because there are so many mutations in the human genome, what we're looking at in the rare mutations thing is what are called de novo loss of function mutations. So that there are mutations that mom and dad do not have. They happen somewhere in the embryo, and they cause the gene to no longer have function. And that specific type of mutation is less common and therefore easier to assign to a specific characteristic. But to answer the question, that's something that happens very early in infancy. It's not something that happens after birth.
Take another one from the Q&A and then we'll see if there's another one. Pertaining to the difference between male and female incidents, could it also be that symptoms present differently, are interpreted differently between the sexes? Example, the way the feelings are expressed, explained?
Yes, that is, sorry, repeating the question, which is, I'm going to, well, yeah, I'll just repeat the question. Is it possible that the difference between diagnosis of males and females is just a difference in expression of autism that gets picked up more often in males? And the answer is absolutely yes. It's entirely possible that there is a severe underrepresentation of females in what is really autism that just doesn't get picked up because of social norms or because of what is often referred to as the protective effect of having 2 X chromosomes. That it is entirely possible that that sex skew is because we don't understand autism well enough and not because there's actually a sex skew. Any other questions in the room?
Yeah, I was just wondering, obviously there's different genetic ways that you can get similar phenotypes for a lot of things. And so is this going to be a case, do you think, where there are so many different genetic variants that lead to so many different ways that people have autism? Is it going to be difficult to pinpoint, all of the different variants, do you think?
I think that, okay, so the question is it going to be something where there are so many different genetic variants that lead to so many different types of autism that it's going to be difficult to nail it down? I think that's true, that there are certainly specific genes that give specific types of autism, like the CHD8. Those kids have typically have, so there's again 20 of them. So if we're able to look at them as a pool, they tend to have bigger heads. They tend to have GI problems in addition to their autism, and they tend to have lower IQ. So that's a subset of kids of autism. But yeah, I think eventually that's what's going to happen, is that much like cancer, right? So cancer doesn't mean anything anymore. Cancer, there are literally hundreds of different types of cancers, right? And each one of them has a different therapy, has a different cause. That's going to be true, I think, for autism and schizophrenia and most of what we think about as mental health issues as well. There's certainly going to be genetic factors that contribute to subsets of autism. There's also going to be, I think, environmental factors that contribute to other subsets of autism. Yeah.
This is, I'll have you repeat the first part.
Okay.
So my daughter has both Ehlers-Danlos and autism. And I've seen some studies showing some increased risk of autism with that syndrome.
So the question is, my daughter has Ehlers-Danlos. Ehlers-Danlos. It seems like, so the question is whether or not that has a higher risk of autism.
Says it needs to be studied better.
Needs to be studied better. Okay, so I would agree with that, because I don't recognize the name of the syndrome. Okay. So I will write it down and make sure that I look into it, because I don't know enough to answer a question about whether or not there's higher risk of autism.
So then the actual question is, wondering if there are studies showing more correlation between other conditions and autism and other health conditions. If so, do they think that there's more gene or environment related? Right.
So the question is, are there correlations between other conditions with autism and other genes with autism? And the answer is yes. So there's an interesting correlation between autism and especially like ADHD, attention deficit hyperactivity disorder, where the genetics seem to be similar. And there's also a lot of overlap of kids who have both autism and ADHD at the same time, in ways that I think people recognize as being true, but don't understand why yet. So that's not a fabulous answer, but I do think there's going to be several of these types of syndromes that overlap with autism, and eventually we're going to figure out that they're similar mechanisms.
One more from online. What testing should a parent with an ASD child be asking for from their physician?
What testing should they be? So this is not my expertise. They should be asking for a clinical diagnosis. And one of the good and bad things about autism is that you need to get that clinical diagnosis, and then you need to get to the services that your kid needs to help them. And a medical doctor, frankly, is not that useful. Medical doctor can't do a lot for your kid with autism. If they have language impairment, they need to go see a speech pathologist. If they have trouble with, the basic things in their life of getting around on the streets or tying their shoes, they need to go to an occupational therapist. There's the medical doctor is not very useful. So what you need from your medical doctor is a reference to go get a diagnosis, and from there you need references to the treatments that will help your kid.
So I'm kind of curious, let's say, we knock down or we silence those genes contributing to autism. Would there be like a negative or an unexpected effect on the body? Because I know you're like, I would be like shutting down or like silencing the entire gene or like certain parts of it. And there's like a bad effect on the entire body.
So there's certainly a bad effect on the, yeah, oh, sorry. I will learn. It's almost over. So the question is, if we knock down the genes, I assume we mean specifically in the brain, but if we knock down those genes all over the body, are we going to get outside of brain problems, right? Yeah, and so the answer to that is certainly yes. So part of the reason why we did the mouse studies is I showed that there's changes in metabolism and fat content and how much the mouse weighs that have nothing to do with the air pollution impacting their brain development, right? But it's clearly impacting the entire body, and that's also true of all these genes that we found that have a link to autism. That lots of kids with autism who had mutations like this, it's not just their behavior that's different. It's also they have GI problems. They have problems with gait sometimes. So it's certainly not restricted to the head for sure. Any other comments or questions? Oh, one more.
So autism and it can show different people. Has there been research to determine whether a specific gene would correlate with a specific symptom or manifestation of autism?
Right. So the question is that I talked about there's different types of autism, and I've talked about there's different genes that cause, and the question is there a correlation between having certain mutations in certain genes and having different symptoms of the autism? That okay? And yes, absolutely there is. So one example I gave is the CHDH gene. Those people who have that mutation also have gut problems, they have bigger heads, which is not all that uncommon in autism, but it's every one of these kids has a bigger head. They also have problems with gait, and so it looks like there's a specific syndrome. If you have CHD8 mutation, this is your type of autism. And then the rest of autism we haven't explained yet, but we might end up with similar subsets of autism where if you have a mutation in this gene, you've got this type of autism, and hopefully, like cancer, we can get better at treating those specific types of autism, right? So there's not a cure for cancer, but there are therapies for each of the hundreds of different types of cancer so that the survival rate is much better than it used to be. And eventually we'll get there for autism and other mental health concerns as well.
One more question online. Is the correlation between autism and air pollution consistent with the rates of autism being higher in geographical areas where pollution is higher? Has that been studied outside of the US?
Oh, wow, that's a great question. The short answer is yes to higher rates of autism, but there's other factors that can't be accounted for, right? Higher air pollution is in bigger cities. Bigger cities have not only more people, but also generally have better medical care where the kids actually get diagnosed with autism. So the short answer is yes. The longer answer is it's not entirely clear. Looking outside of the US, yes, there are studies that have been done in Europe, and they get essentially the same results, where air pollution is the way they do it is to look at severity of symptoms, and definitely air pollution is correlated with more severe symptoms of autism. So. Oh.
Okay. Well, thank you, Dr. Campbell,
Yes.
for being here, and round of applause. Very much appreciated your talk and
Yes.
you sharing your research. Thank you for all of you who are here in the room tonight, and for those tuning in on Zoom. We appreciate you being with us. And stay tuned for more coming from your health lecture series throughout the year. So be sure to check out the website for more of the monthly opportunities. Thanks so much.
Thanks, everybody. You're awesome.